Today we're going to learn more about genetically modified organisms as we see how something called recombinant bovine somatotropin, or RBST, is made. RBST is a hormone that increases the amount of milk that a cow can make, and it's actually injected into a whole bunch of cows in the United States, and these cows are used probably to make the milk that you drink. By the end of today's lesson, by watching these videos and doing the class activities, I want you to be able to do these 4 things. First, list and describe the steps needed to insert a gene from one organism into another organism. Today, we're going to go from a cow into a bacteria. I want you to be able to explain what restriction enzymes are and why they are used in making genetically modified organisms. I want you to be able to explain what DNA ligase is and why it's used to make genetically modified organisms. And finally, I want you to be able to tell me about what RBST is and what it does in cows. So here's a quick review of the steps to make a genetically modified organism. If you remember, first you have to identify what you want your organism to do. Then what you have to do is find another organism that does that and take out the piece of DNA or gene from that other organism that's responsible for that trait. Finally, you need to take the gene out of the one organism and put it into another organism. Let's go through these steps with how RBST is made. First, what's the desirable trait of the organism that we want to do? We want to make bacteria, but what we want to do is reprogram the bacteria so that they make this hormone called BST that we can inject into cows. Before we get too much into this, I want to give you some background information about what BST is and what it does. So, BST stands for bovine somatotropin. And what it is it's a growth hormone. This growth hormone is normally made by cows, but how farmers can use this as a tool is that they can inject BST into cows to make those hormone levels even higher. Now, the type of BST that's natural is just called BST. The type of BST that's made in a lab is called RBST. And the R stands for recombinant. And all that means is that you've switched around the DNA or did some cutting and pasting like we'll do today to make this artificially. So, bacteria are used to make RBST. That RBST is isolated from the bacteria, or taken out of the bacteria, and injected into the cows. Now, what happens to a cow that's injected with recombinant BST? That causes the cows to eat more food, and it causes that food to go to the mammary glands where the milk is made, so that the cow produces more milk. And it's estimated that a cow is going to produce about a gallon more milk per cow per day. And normally, a cow makes about 7 or 8 gallons of milk per day, so this is going to boost that up by about 15%. Let's blow up this graph over here to take a closer look at what's going on there. So this graph has a lot of interesting data, and it's actually graphing 3 different things. Milk production, the amount of milk each cow makes, and how many cows there are. And all of that is graphed against time. So this data shows from 1985 to 2010 and beyond, so a period over 25 years, what happened. So what you can see over these 25 years is that milk production increases. and it increases from about 150 billion pounds to about 200 pounds. Over the same time period, the output per cow, how much milk is made by each cow, increases as well. And to put a number to this, the output per cow increases from about 13,000 pounds per cow of milk to about 23,000 pounds of milk per cow over those 25 years. Finally, the number of cows actually goes down over this 25-year period from about 11 million cows to about 8 million cows at the end of the 25 years. So what is this graph telling us? Well, it's telling us that the milk production is increasing, but the amount of cows go down. So what that means is the amount of milk that each cow is making is going up quite a bit. How is this possible? It's possible because more and more cows are being injected with RBST. So now that you know a little bit about what RBST is, hopefully you understand why farmers would want to use it. Because they can increase the amount of milk that each cow makes, and they don't have to keep as many cows around on their farm to get the same amount of milk. So, what's the desirable trait? We want bacteria to make BST so that we can get it and inject that hormone into cows. The next step is that since cows make BST, they have a gene to code for BST. We have to get that gene out of cows. Then finally, we have to put that gene into bacteria. Once the BST gene is in bacteria, the bacteria can be grown in a lab so that the bacteria are now reprogrammed to make BST, which we can then take out of the bacteria, put into a syringe, and inject into the cows. So in today's lab, we're going to simulate this part with paper. We're going to be doing steps 2, 3, and 4 of this. Yesterday, when we isolated our own DNA, we were simulating what it would be like to do this step. Today, we're going to complete the last steps. Before we jump in, it's important to know what tools we're going to need to make our recombinant DNA, or RBST. We're going to be reprogramming bacteria, so an important tool that we need is the bacteria themselves. And we're actually going to be using a piece of DNA from bacteria called a plasmid. Another tool that we need is called a restriction enzyme. So restriction enzymes are like scissors, just like shown in this picture here. What do restriction enzymes do? They cut the genes out of DNA. So what we're going to have to cut, first, we're going to have to cut the RBST out of cow DNA. And the second thing we're going to have to do is we're going to have to cut open bacteria DNA so that we can put that BST gene in. The another tool that we'll need is called ligase, and ligase is an enzyme that's like glue, and it pastes pieces of DNA together. So after we cut out that BST gene and we cut open the bacteria plasmid, we need to glue them together with ligase so that the bacteria can be reprogrammed to make BST. So, I want to take a minute to draw out these steps together as a class. So after you watch this video, we're going to put together all these steps in our notes as a class. Hope this was helpful.